Thank you so much for such a kind introduction, Veronica. It's really fantastic to be here today with you all. Thank you so much for this invitation. Yes, so I just wanted to start with a brief personal introduction. And this is just kind of going over a little bit of what Veronica said, but I am a Nova Scotian. I was born in Halifax and I grew up in Cape Breton. And I actually had an original career in souvenir wholesaling. I actually started off as an entrepreneur and I had a corporation by the time I reached my undergraduate studies, and it was through this that I helped to finance my studies. I am a professional graphic designer. I studied at the Art Institute of Toronto. But really, my research career began, as Veronica said, in her lab studying the lace plant and programmed cell death. Veronica is my mentor and my friend, and she really opened my eyes to the world of research, and my passion for research was found under her tutelage. And yeah, currently I'm splitting my time between SLU, Sweden, and the University of California, Davis. I'm actually going to UC Davis next week. And SLU is the Swedish University of Agricultural Sciences, and I live and work in Uppsala, and this is their main campus. This is really a research-heavy institution. The staff to student ratio is 2 to 3, so it's really like a core research facility for Sweden. Yeah, the support from NSERC is what initially brought me to Sweden, and since then, I've received funding from Formus and Carl Triggers primarily. Yeah, so as Veronica mentioned, I started my research career studying the lace plant, or Patagonia madagascariensis, and this is a beautiful model system. And it's interesting because it has this unique form of developmental PCD that's very rare in plants. Another one that you might have seen before is Monstera obliqua, which is pictured over here. Yeah, and it was in Veronica's lab in her live cell imaging suite where I really had plenty of access to wonderful microscopes. And me being someone interested in graphic design and visual science, microscopy just was a really great natural fit. And I spent a lot of time during the PhD trying to automate things and processes, just being able to collect more and more data more efficiently and translate that to understand biological phenomena. Since leaving the lace plant research primarily, I've been, like I said, living in Uppsala, Sweden. This is a city, quite small city, just north of Stockholm. And it is famous for having some of the great scientists in Swedish history, including Carl Linnaeus, who is the father of modern taxonomy. Culturally, Uppsala is at the heart of Sweden. It is the site of the Viking burial mounds, and it has Scandinavia's tallest cathedral. And yeah, being so close to Stockholm is excellent too. Stockholm is a beautiful city with lots to explore, and it's only about 25 minutes by fast train. Okay, but now I want to get to the core story. This is my main research project, and it involves the use of chemical genetics to understand plant autophagy mechanisms. And if you're unfamiliar with chemical genetics, essentially it is a biochemical approach wherein we use small molecules to target and identify protein targets. And this is often done through biochemical screening. And autophagy, or self-eating, is a fundamental biochemical process that has such great significance to all eukaryotic organisms that the initial pioneering work done by Yoshinori Sumi in his yeast systems was awarded the Nobel Prize for Physiology and Medicine in 2016. So first, I'm going to explain autophagy in greater detail. So autophagy, or self-eating, is a fundamental biochemical pathway wherein cargoes within the cytoplasm of cells are targeted for degradation by double membrane vesicles known as autophagosomes. So these vesicles will initially start out as a small phagophore assembly site. They'll begin to round up cargoes and expand. They'll fully engulf them. And then they'll be trafficked to the vacuole, which is the lytic compartment of the plant cell. And then the inner membrane is released into the vacuolar lumen, and then it is degraded by the hydrolases and the low pH conditions. And this allows for the recycling of components that are no longer needed. So autophagy is very critical. It's our major recycling and degradation pathway in all complex life forms. And so you can imagine if autophagy is impaired, there's some pretty significant impacts with that, because if there's, let's say, damaged components that can't be removed efficiently, they're going to cause damage to other cellular components and lead to possible mutations, et cetera, cell death, and it can be really devastating. I labeled here in this diagram ATG8. This is autophagy-related protein 8. This protein is actually very important for studying autophagy because it labels both the outer and inner membranes. So it's decorating the autophagosomes, and it also gets delivered with the inner membrane for degradation. So it's a protein that we can use to track autophagy processes from beginning to end. And this will be important when I explain the labeling schemes and how our assays work later on. So as I alluded to, autophagy is a fundamental biochemical process. For a long time now, we've known that, starvation-induced autophagy can affect lifespan in animals, for example, but it's also very important for plants. Some recent studies have showed that enhanced autophagy can increase growth, it can increase stress tolerance, lead to higher fecundity, so greater seed yield, and increase longevity in plants as well. So the opposite is true in general with deficient autophagy. And while this has been studied in laboratory settings and model systems, we have yet to translate this fundamental knowledge to agricultural applications, but it's a point of great interest and significance right now. So the significance of autophagy modulation is quite important because it gives us this viable pathway to having ways to improve crop production. And if we think about, hurdles and actual practicalities, there's a pretty stark contrast between use of chemical modulation or the use of genetically modified organisms. And there's also a problem not just with political applications for GMOs, especially here in Europe, but with GMOs, you're kind of forced into having constitutive autophagy upregulation or downregulation or inhibition, and that just causes a lot of problems because autophagy is a fine-tuned process. So I'm mostly interested in chemical modulation of autophagy because with this we can have precise targeted applications. And there's a problem that exists right now in that available chemical modulators of autophagy have off-target and pleiotropic effects. So the chemicals that are currently available tend to cause a lot of stress and damage within cells, and they don't specifically target autophagy. So we have this urgent need for new autophagy modulating compounds that can do so with low toxicity, but good specificity. And there's an additional benefit to studying autophagy and finding new autophagy modulated compounds in that new compounds can be leveraged to further unravel the autophagy signaling mechanisms in plants, animals, etc. Yes, so this, I just want to highlight too, the challenges of autophagy modulation have actually been reviewed recently by some previous collaborators of Veronica and me that have looked at the existing modulators and shown that most studies, individual studies, don't well characterize autophagy modulators, and they have a tendency to fail in clinical trials. So when you're trying to use autophagy modulators in cancer cell lines, for example, this tends to fail. There's none that have been successfully implemented. And they highlight the need for these specific non-toxic modulators. So in what was it, 2019, we kind of finished up our initial chemical screening pipeline for identifying plant autophagy modulators. And the overview of the screen looks like this. We have 4 phases. The first is hit identification, and this is a dual luciferase assay. The second phase is hit optimization, and this is with our tandem tag assay. Phase 3 is then a confirmation using a canonical well-established method, the GFP cleavage assay. And then phase 4 is a specificity check using endomembrane marker lines. And just some general notes about the screen. We go from higher throughput and lower reliability to higher reliability and lower throughput methods. And in this talk, I'm going to talk about each phase and give you the details. And I have schematics for each one, which we will go into. Essentially, each one uses a different type of tag. And as I mentioned earlier, the ATG8 is important for labeling our autophagosomes. We also use NLS tags. These are nuclear localization signal tags. They vary in terms of the methodology. The first one is the high throughput chemiluminescence assay. The second, the tandem tag, is a confocal microscopy assay. The third is a Western blotting biochemical assay. And the phase 4 is another confocal assay. So I will go into each of these now, each phase, and describe the story. So the first phase was hit identification. And for this, it is a dual luciferase assay. So we use 2 different luciferases, Firefly or FLuc, Firefly luciferase, which was fused to ATG8, or Ranilla luciferase, RLuc, fused to NLS. This is that nuclear signal, or FLuc fused to NLS. And these were stably transformed into BY2 cell cultures in the following scheme. So reporter lines had both FLuc ATG8 and RLuc NLS expressed within them. And what happens here is that when there's basal autophagy going on, the FLuc ATG8, which will be labeling the autophagosomes, ends up getting delivered with cargoes into that lytic vacuole. And here it will be degraded at some basal rate, right? And you can imagine if we have higher autophagy and more autophagosomes being labeled, you'll have more turnover and degradation of that FLuc ATG8. So eventually you get, with autophagy enhancement, a higher RLuc to FLuc ratio, because that RLuc is going to be protected within the nucleus, where the FLuc is going to be degraded. And in our control scheme, we have the RLuc and the FLuc both protected within the nucleus by NLS, so it doesn't respond to autophagy modulation. So this is the way that the first phase was set up, and this was done in BY2 cell cultures in a 96 well plate format. And we did 24-hour treatments of our compounds. And we started off this drug screen with a smaller set of compounds, but this was a pre-screen library that my collaborators from UC Davis actually had narrowed down from 20,000 compounds. And they suspected that they influenced the plant endomembrane system, but they didn't know how exactly. So it was a good candidate group to start with to look for autophagy modulating compounds. And this work was carried out in collaboration with the CBCS, which is the Chemical Biology Consortium of Sweden. As I mentioned, it was done in tobacco BY2, which stands for bright yellow 2 suspension cultures. And these are developed from CalI. So these are really, it's a nice model system to work with because it's very easy to transform with agrobacterium tumefaciens methods. And they're really easy to maintain. And if you haven't seen callus induction before, this is coming from some leaf explants. Yeah, so back to the data. So with this system, we carried out the high throughput screen and we identified 19 enhancers, 10 of which were strong enhancers that had greater than 3.3-fold increases in that R-loop to F-loop chemiluminescence ratio. And we also selected 5 inhibitors of autophagy that had greater than a 2-fold decrease in the R-loop to F-loop ratio. So going from phase 1, we identified some hit molecules, 24 to be precise. And in phase 2, we wanted to move just from general autophagy flux changes to confirmation of cargo delivery. So making sure that the cargoes are in fact being delivered to the vacuole. And for this, we have the following scheme. We developed a tandem tag confocal microscopy assay. So with this, we have a red fluorescent protein and a green fluorescent protein, both tagged to ATG8. And the way that this works is we have this tag expressed in both a wild type Arabidopsis background and an ATG5, ATG7 knockout mutant control line. So in our wild type reporter line, the tandem tag would look like this, where it labels the outer and inner membranes of the autophagosome again, and it will get delivered to the vacuole. And the vacuole here is red because in the acidic environment of the vacuole, the green fluorescence protein, it is actually quenched. So the green fluorescence turns off in our confocal microscopy scans. So you end up getting an accumulation of red signal when you have autophagy upregulation. But in ATG5-7 knockout line, you can imagine, okay, yeah, nothing happens. The tag is just there in limbo. It's not being delivered to the vacuole. And so our readout here is just the red to green fluorescence signal in the vacuoles. And the way that we developed this is, so it's a confocal microscopy assay. It's lower throughput compared to phase 1, but it's really powerful in that it also gave us morphological data. And this is what we utilize for our confirmation and optimization of the chemical treatments. And just here's another scheme of a tagged autophagosome. And so when it gets delivered, you end up getting that red accumulation within the vacuole, right? And this is the Arabidopsis seedling. This is, we actually do the scanning in the root, and we focus on the differentiation zone and the epidermal cells. This is where we can see the vacuole most clearly, and we want to avoid scanning in the root tips in this case because it obscures the signal because there's lots of dying cells there, and meristematic regions are, in general, the expression of our tag is too strong, and it's not as reliable as looking at this differentiation zone. And what we do is we take seedlings that are grown first vertically on MS plates, then we transfer them to 6-well plates such as this, and then we treat the specimens with the compounds or vehicle control, and we have both our tandem tag control and tandem tag reporter lines simultaneously. Then I would run semi-high throughput confocal microscopy. So with a motorized stage, we can pick the root regions of 6 seedlings at a time, no problem, and tell the microscope to scan several areas within each differentiation zone, and then we just let the data get acquired automatically. And then we developed an automated image analysis software for statistical analyses. So we had our tandem tag control reporter lines, and we set out first by doing some viability testing. We tested a wide range of each compound. We went from 0.05 to 200 micromolar, just to get a general sense of which concentration range we wanted to be working in for the confocal work. And the treatment applications were then optimized for time and dose in that differentiation zone. And the data that I'm going to show in the coming slides represent 37 experiments, 2,000 seedlings, 6,500 micrographs, and about 20,000 individual vacuolar measurements. And you can imagine why we needed the automated image analysis and software. It's because these large data sets were just too cumbersome to work with. When I first started running these experiments, it was manual quantification, just using the DIC channel. And I would just pick, 3 random vacuoles and get the RFP to GFP ratios. And I get something like this, where I see, okay, a response over time by different doses, et cetera, for each compound. But yeah, because of the size of and scale of the data, we developed this software, which is just using free Fiji or ImageJ and RStudio. And the nice thing about our confocal platform here is that everything's connected online to a server. The files are backed up automatically. And essentially all what I had to do after scanning was come back to the office and run the software, and I would just select the scanning folder from that day. And then you'll see as it runs, it just automatically selects the vacuoles, and then it creates a data table giving you the RFP to GFP ratios and generates the charts for you. So this was a major upgrade and takes out any type of bias that could be involved, but still, like, comparing my manual quantifications to the automated, it was matching up quite nicely, which was great. And to make this software more user-friendly for people who would be interested in using our tandem tag assay, we also developed a calibration macro. So ideally, this will work for any type of confocal microscope data that you collect too. So the nice thing about Fiji is that it's friendly for like Nikon, Zeiss, Olympus systems, etc. And yeah, so the software is openly available on GitHub, and we've published a follow-up methods paper calling it Autoflux, which is out and available. Yeah, so back to the main story and the main data set now. So here we have our reporter line, and in our vehicle control, the seedlings, the vacuolar region would look something like this. So they're mostly just dark, right? And with AZD, this is a positive control. That's a known enhancer of autophagy. You would get a buildup of red in the vacuole. Something similar happened with our C12. And in our knockout control tandem tag line, you don't have that same kind of response happening as expected. And so we used these experiments to kind of pick which compounds made sense to work with moving forward. So we're looking at which ones had a good increase in terms of autophagy upregulation and cargo delivery, and didn't have any kind of influence in the RFP to GFP ratio in the control knockout line. Because if you do have a response there, then it's probably telling you that there's something wrong and that the compound could be very stressful. Yes, so from this data, I mentioned before that the nice thing here is that we're getting morphological data as well, right? And I'm just going to show you our autophagy inhibitor. And for this, we stress the seedlings with minus nitrogen starvation beforehand. So even in our vehicle control with minus nitrogen, there's been an upregulation of autophagy from that starvation, and the vacuoles are red as expected. But with our inhibitor, we saw this where, oh no, the vacuoles are still quite dark, and it looked like there was a lot of puncta autophagosomes being built up within the cytoplasms. And at lower concentration, that kind of effect diminished in terms of just the number of puncta being observed. But still, we didn't have red vacuoles, so the inhibition was still working. And here's just a video showing that high concentration of inhibitor 4, because it kind of nicely shows the power of having morphological data to go along with this. It looks like things are kind of blocked and jammed up within the cytoplasm here. So there's a good reason to suspect that this inhibitor is actually a fusion inhibitor. It's stopping the delivery of the autophagosomes to the vacuole. Yeah, so from phase 2, the confirmation of cargo delivery, to phase 3, we carried on with 4 compounds. And this is the verification of flux completion. And by that, I mean the final degradation of cargos, making sure that that is occurring. And for this, we used a very well-established assay known as the GFP cleavage assay. And for this, what we have is just a green fluorescent protein fused to ATG8. And that is expressed stably in both a wild type reporter line and an autophagy deficient control background for the control. Yeah. So the wild type reporter line is shown here. So with this, the GFP tag will get delivered to the vacuole. And here what happens is that the regular degradation pathway and breakdown of the products will cause the breakup of GFP from ATG8. And so you end up having free GFP cleaved from the ATG8. As well as you'll still have some degree of your fusion protein as well. And in the knockout control, you don't see the same kind of process happening. So this is a Western blotting assay in which we quantify the cleaved GFP to total GFP ratio as a readout of autophagic flux. And this was done in Arabidopsis, and we tested whole seedling versus differentiation zone only, and using just the differentiation zone is quite a bit better than using whole seedlings. So I'll just show the data for the differentiation zone. So the GFP cleavage assay looks something like this. So we have our vehicle control treatment versus the AZD. This is a positive control. And as you can see here with this AZD in the wild type reporter background, you have this increase of free GFP versus the fusion protein up here. So this is the GFP and ATG8 together up on this line, and we have just cleaved GFP down here. And so we performed the same kind of experiments, but with our vehicle as well as one of our compounds, which I'm just showing you C12 here in both that wild type background and our autophagy mutant background. And you see that there is an increase in the free GFP on C12 treatment, and there's a decrease in the fusion compared to our vehicle control. And in the ATG5 knockout, we don't have that same kind of response. So this one is an example of a compound that cleared our phase 3. And from there, we move on to phase 4, and this is a specificity check. And I don't have a schematic for this one because it is simply a collection of library of Arabidopsis endomembrane marker lines, which included 17 different fluorescent tags to various endomembrane components within Arabidopsis. And with this, we tested a gradient of the selected compounds to see which deleterious effects, if any, would occur to any endomembrane marker lines. And for this, we did just manual phenotyping of over 1,000 seedlings. And the example that I'm showing you here is HDEL-GFP. So this is a marker for the endoplasmic reticulum of plants. And so you have this nice reticulated network. And then in Arabidopsis, you have these ER bodies, which are these things that you see here. And with compound 12, for example, at 10 micromolar, the network looked really good. At 30 micromolar, it still looks quite good, but at 60, 120 higher concentrations, in some cells, you start to see signs of stress, where instead of having the nice network, there's these kind of blobs and aggregates that start to form. And yeah, so this was also very useful because when you're looking for phenotypes at the cellular level with new compounds that you don't know their targets for, having an idea of which endomembrane system components are being affected can help you identify the targets downstream. And yeah, so for the confocal data and the phenotyping, we did both time series and Z-stacks on the seedlings so that we could have a good representation and an idea of what was happening within the cells. And here again, we see like healthy, good conditions for the lower concentrations of compound 12, but at higher concentrations, there's some signs of stress starting. And this is an early version of what we're calling the phenotable. There were 17 marker lines, and for each one, we were like making notes on what we saw with the various scans after running multiple replicates at various concentrations. And even something that we noticed early on was some of our control drugs, even though that we were using them at concentrations quite a bit lower than the literature suggests we should be using them at, and quite short times, we're seeing dramatic phenotypic changes. So it kind of reinforced our thoughts on the need for better and alternative autophagy modulating drugs. And yeah, so here is just a summary figure from some of the phase 4 data where we see compound 12, for example, working quite well at 30 micromolar and below in terms of GFP cleavage assay and our tandem tag, but not having any dramatic influences on the endomembrane system with our specificity checks. So ultimately, this screening, chemical screening pipeline was published as a breakthrough technologies paper in plant physiology. And in summary, we developed this new pipeline to systematically screen for plant-specific autophagy modulators. And through it, we identified several new promising small organic molecules that we saw influence autophagy in tobacco, BY2 cell cultures, as well as Arabidopsis thaliana whole seedlings. And the next phase of this project has been moving towards drug target identification. And so the work that I'm showing from this point on is unpublished. The mechanisms of the new autophagy modulators are really important to identify. As I mentioned in the introduction, part of the search for new autophagy modulators, the benefit of having new tools to modulate autophagy, it's not just for direct applications, it's also so that we can use them to try to find their targets and learn more about the autophagy signaling pathway. We still don't know a lot about how exactly we can modulate autophagy in all the various ways. And so that's what this phase of the project is working towards, finding those protein targets so that we know more about autophagy signaling and have a new target to work with. And for this, there's 4 main phases. First was developing a library of analog compounds. Second was determination of the structure-activity relationships of the compounds, and then adding a probe with the help of the CBCS again, and then running and developing an in vivo pull-down assay. So first off was development of this analog compound library. And we used PubChem to look for structures that were chemically similar to our parent molecules. And ideally what we were looking for was maybe 1 or 2 small changes from the parent molecule. And then we searched and tried to acquire whatever we could through commercial means, and what we couldn't find but really wanted, we had the CBCS synthesized for us. And then the second step was determination of structure-activity relationships, or SARS. And this was done with 7-day-old seedlings. And we used the tandem tag autoflux protocol. And for this, we had somewhere around the order of 10,000 measurements done. So we used that tandem tag assay and we scanned the seedlings. And yeah, we we're trying to compare the analog compounds to the parent molecules at several different time points. I'm just showing some time point 3 data on this slide, but the important thing is that we looked at the how the structural changes influence the autophagy response, and that allows you to understand which area of the molecule is available for labeling and modification. Doing that, we found which regions of the chemicals were not responsible for the autophagy response, and then we did chemical synthesis of the probe with the CBCS. So these new probe molecules had the addition of diazarine, which is a compound that if you hit it with UV light in the 350 to 380 nanometer range, it converts to a highly reactive carbene, and this will form covalent bonds with proximate biomolecules, i.e. the target protein, if everything goes according to plan. So our autophagy modulator probes look something like this. It has the pharmacophore, so this is the portion of the molecule that's responsible for the active response, the autophagy influencing response. Then we add the diazarine probe, and we had an alkene handle on this end of the probe so that we could do downstream click chemistry to add biotin. And in our control scheme, instead of the pharmacophore, we just had a phenolic ring fused to the probe, which also was available for click chemistry and biotin addition downstream. And once we got these chemicals synthesized from CBCS, then we tested them with autoflux to make sure that the autophagy response was still intact, because modifying the structure and adding a probe to it could have easily caused issues with either solubility or engagement with the target, or reduce the response to the point where we couldn't use it to induce autophagy anymore. But we did get good autophagy responses still with 2 of the probes, C12 and C7. And we then worked with the probe molecules comparing to the control and tried to find a time point where we were getting good autophagy promotion, and we saw higher amounts of autophagosomes being produced. So then that was a good time point to run our photoaffinity labeling assay, which is the stage 3, the third, it's the final phase of this part. So the photoaffinity labeling assay, or PAL, looks something like this, where we have our treatment done in whole seedlings, and the pharmacophore with the inactive photoreactive probe is supplied to the seedling. Then we activate the probe with UV light, and then we lyse the cells, and we do click chemistry to add biotin to the probe, and then we do affinity purification. We use streptavidin magnetic beads. So streptavidin and biotin will bind very strongly together, and so we use these magnetic beads to fish out our labeled and captured protein. And then we were running STS page analyses to see if we were getting differential capture between our various conditions, and using that to optimize this whole workflow. Because this was a new assay that we were developing from scratch, essentially. And then the final step is to run affinity enrichment, LC-MSMS, to identify captured target proteins, and we have our control probe compared to our pharmacophore probe. So in general, so we had our treatment in vivo of our seedlings. We optimized time for treatments for both the probes that we used. Then we did our probe activation with small handheld UV light. And then we would do protein extraction, pretty standard methods here in general. And then we performed click chemistry. This took quite a bit of work and trials trying to optimize the conditions. because we were playing with the various components, and yeah, this took some time. And we also had a problem wherein we had excess biotin that was kind of binding to our beads downstream, so we had to work with Zeba desalting columns to get rid of that. And then we would do purification with streptavidin magnetic beads, like I mentioned. And so we worked with those conditions, and we were detecting with STS-Page. For this, we used Oriole staining, which is a product from Biorad, and it's more sensitive than your standard like TGX stain-free labeling, which is in the pre-made gels these days. Yeah, and essentially after working through those protocols, developing that, we would send samples for proteomics, but we would split our samples to do like a gel verification step, and then have the remaining sample, which we would send on to the proteomics facility. And just to show you, like after optimization and developing this assay, we were looking for something like this, where we had clearly different capture that was happening between our pharmacophore probe and the control conditions. And the facility that we did the proteomics work with was at VIB in Ghent. And this was supported through a grant that I secured with Epic XS, which has the Horizon 2020 backing. And yeah, so with this, we were able to compare the captured proteins in our modulator probe versus the control probe. And the data that comes out looks something like this. We were given volcano plots and a list of top hits, which were upregulated within our pharmacophore treatment group versus the control probe. And these are log2 fold change values. So it was, for the higher hits, it was in the order of 30 to 50 times higher levels than even imputed control values. And the two leading candidates for C12, for example, happen to be proteins from a single family, and they are quite interesting. We believe that they're linked to metabolite damage control in some way. I won't be revealing the actual targets here today, but this is what we're working with now. And our current efforts are now geared towards validating these hits. And to do that, we're taking an approach, several different approaches, I should say. First is the production and purification of his-tagged proteins. So we're adding a polyhis tag at the end of these proteins, and then this will allow us to do several experiments, such as the cellular thermal shift assay, or CETSA, and we can also do microscale thermophoresis. That's done in collaboration with the Drug Discovery and Development Platform of SciLife Labs here in Sweden. And then we're going to run biochemical assays whenever appropriate to do so. It depends on the target that we have, right? What biochemical assays that we'll run. But I have a scheme here just for CETSA, because it's kind of important to understand when this kind of thermostability assay is fundamental in drug discovery these days. So essentially you can do protein extractions or whole plant seedlings, or work with lysates, and what you do is you treat them with your small molecule or your vehicle control, and then you aliquot after X amount of time of treatment into various, we use PCR tubes and use a thermocycler at various temperatures, so that you get a gradient of different heat treatments to those conditions. And essentially what happens is, when you have a drug or a ligand that binds to a target protein, it confers thermostability. So it will be less likely to aggregate and be pulled down when you do, even just basic centrifugation steps. You'll maintain more soluble protein within your soluble fraction when you have binding versus when you don't. So you end up having this kind of thermal shift in terms of at what temperature will the protein aggregate and be pulled out of solution by centrifugation. And this is one way that we commonly identify targets these days. Microscale thermophoresis works in a somewhat similar way. It is a much more sensitive technique that uses these microcapillaries and infrared laser. And essentially what happens is if you have binding, there will be slightly different movement speeds of your compound within these capillaries after heating with the laser, and the device will automatically track these curves, and then you can test various compounds and figure out whether or not you have binding of your target to your ligand, your drug. And yeah, so we're running these two assays primarily, and these are preliminary results with our leading candidate. But the CETSA, for example, here, this was just done in a lysate, so we have to validate this with purified protein samples, which is underway currently. But essentially, what you're seeing here, this is our Ponsau, so this is our total protein load control. And then we have two different exposure times. But what we saw was that at the 50, 55, 60 degree treatment times, we had thermal protection, more stability of the compound to the protein target. And here's just a clipping of that. So at the 50, 55, and 60 degree, you can see that there's more in the treated group versus the untreated control. We also have these useful analog compounds which have no autophagy response, which we can use in the CETSA assays. And for example, this one, A2, which has very little autophagy response, had no thermal stabilizing protection like the parent molecule did. But as I mentioned, we're still working and developing our target protein purification systems. And for this, we're doing overexpression in E. coli, and then we're using IMAC to isolate the histag proteins, and then using size exclusion chromatography and protein concentration. Yeah, so with these, we're then going to confirm those sets of results, and then we'll be in good shape for that specific target. And in summary, the current progress for target identification, we have these promising small organic molecules. We've developed this novel in vivo pull-down assay for plants, and we have our potential interactors list for C7 and C12, which are autophagy enhancers. And we're currently working to confirm leading interactors using CETSA, MST, and biochemical assays. Just last week, I got some good news, though, that I got some grant support to work with the chemical proteomics platform of SciLife Labs, which is in Stockholm, in a brand new state-of-the-art facility called the Karolinska Institute Biomedicum. And they have state-of-the-art pipetting robots and mass spec devices. And we're going to develop a new assay, a newer assay that's been working, that's been effective in animal systems so far, but we're going to be developing it for plants and use it to identify drug targets. Yeah, so this technique that I'll talk about uses tandem mass tag, or TMT, isobaric labeling. And the scheme here, this is what a standard TMT tag would look like. So you have a reporter ion in a balancer group, and it labels specific peptides within your samples. And it requires tandem, so two rounds of mass spec. So in the first round, you'd get something like this, where you have, in this case, there's 11 different tags that were utilized. And then, so an individual protein, for each sample, like you'll be able to determine it, like this is one single peptide, for example, but from all the different all the different input samples. And then through the second round of mass spec, you actually can determine the relative abundance of an individual peptide, so an individual protein, from each of your input samples. So it's a multiplexing mass spec technique, which is pretty widely employed these days. But the new assay has kind of elevated this this use of TMTs and combined the same principles as the CETSA and other thermal proteome profiling techniques. And it's called the PISA assay, the Proteome Integral Solubility Alteration Assay. So this is a high throughput method that essentially you model the loss, the degradation and loss of soluble protein over the temperature gradient. So same kind of principle as the CETSA, but that's within a single sample that is then multiplexed with others, and you can detect your target proteins and drug interactions this way. So it's like combining mass spec and CETSA together. So essentially the workflow looks like this. You treat your cells with vehicle or your compound, and then you split into temperature groups, just like CETSA, but then they're pooled back together. And then you do ultracentrifugation, you collect the supernatants, you do peptide digestion, then you label with your TMT, your isobaric label, and then you can, that way you multiplex, and the system is up to 16 TMT tags at once. So if you can imagine this, in my condition, I can use the parent molecule in 3 replicates, 2 analogs, a positive control, and you know, an untreated sample or something to reach my 16 sample input altogether. Which are then fractionated and then run through standard LCM SMS methods. So that way, all in a single run, I can see which proteins were protected with the binding of my drug to the target, and which you know, which proteins were not. And yes, so the that's like one-dimensional PISA assay. I think that's enough information on that one for now. But this is something that I'll be working with Massimiliano Gattani. He's the platform director here in Sweden. We're going to develop this for plants, which hasn't been done yet, so that they can broaden their user base. Yeah, and I think we've covered that. I think we're getting lower on time too. So in terms of the future plans, short-term objectives, we'll be testing compound effects on seedlings, validating that PAL assay, confirmation of the hits being generated, and then establish transgenic lines to unravel new plant autophagy signaling mechanisms. And then eventually we'll do modeling and modifications to the compound structure. So we want to model the binding of the compounds to the specific target. I did want to just talk about like seedling tracking growth development. We actually built a robot system here from scratch, which is open sourced. We call it Spiro, the Smart Plate Imaging Robot. And if you're interested in that, maybe if there's some questions, I can show you some videos of how this works later on. But yeah, I think since we're running out of time, I'd just like to say thank you to all my collaborators. Florentine Ballhaus is a master's student who's about to start a PhD with me. She did a lot of work in this, and thank you again for inviting me here today.